Hello everyone. In this lecture I'll be covering calcium dynamics and signaling. I'll begin by covering resting potentials and how calcium generally enters the cell and where it's stored. I'll then talk about various signaling pathways and proteins that depend on calcium. Then I'll focus on mitochondrial and endoplasmic reticulum calcium signaling and its influence on apoptosis and inflammation. And finally, I'll finish by talking about how calcium influences gene expression. So if you have any questions, please comment below, and thanks for watching. When talking about calcium, we are primarily concerned with its concentration in 4 different compartments. The extracellular matrix, which has relatively high calcium concentration, the cytosol, which has very low calcium concentration, and the ER, right here, the ER has high calcium concentration. And the mitochondria has kind of an intermediate amount of calcium. It's kind of an intermediate between the cytosol and the ER. So lots of calcium outside the cell, lots of calcium in the ER, medium amount of calcium in the mitochondria, and a very low concentration in the cytosol. Now, each of these compartments can experience calcium flux, where some kind of stimulation causes calcium levels to spike. And this is the basis of calcium signaling that we're going to be discussing. The reason we're so concerned with calcium concentration is because calcium binds to thousands of different enzymes, and it has the ability to modulate enzymatic activity, and a lot of these enzymes depend critically on calcium concentration. So when we see calcium flux or stimulation, it modulates a subset of enzymes. So this begs the question, where do calcium spikes come from? How do they work? Well, basic chemistry reminds us that ions and molecules flow down their concentration gradient, and the concentration is highest outside the cell. So our concentration gradient is going to be basically allowing calcium to flow down its concentration gradient into the cell. Since calcium is charged, however, it cannot cross the membrane, so it requires channels. And I'm going to be discussing 5 different channels. So TRPC, which can be seen right here, I have added TRPC, and TRPC stands for Transient Receptor Potential Cation Channel. It's a class of about 7 different proteins that are permeable to calcium, and they are activated or opened by many different cues like GPCRs, G proteins. They can also be activated by receptor tyrosine kinases, phospholipase C, and diacylglycerol. One specific example that involves the activation of these TRPCs is the activation of TRKB receptors by BDNF. So TRKB is the receptor for BDNF, and this recruits phospholipase C to the membrane, where it cleaves PIP2. The resulting diacylglycerol actually diffuses through the membrane. Diacylglycerol will diffuse through the membrane and activate these TRPC 3 and 6 channels. In fact, TRP3 and 6 are likely the mechanisms by which BDNF induces calcium transients that ultimately promote survival. So TRPC channels are really important for the propagation of calcium transients in neurons and astrocytes. We also have NMDAR channels. These are the classic receptors for glutamate. And so these types of channels are technically called ligand-gated calcium channels. And at the excitatory synapse, or even a cell body, you can find these NMDAR receptors that bind to excitatory neurotransmitters, glutamate. And this causes the channel to open and it allows the influx of calcium. The overactivation of these channels leads to a process called excitotoxicity, whereby excessive glutamate floods the cell with calcium, eventually leading to apoptosis. And we're going to discuss a couple of mechanisms by which that could be happening later in the slide. Another important channel are these VGCCs, or voltage-gated calcium channels. They're composed of a central alpha subunit that forms a pore. There's about 15 other subunits that modulate the channel's activity. Generally speaking, the channels open when the cell depolarizes, which only happens in excitable cells like neurons. In fact, VGCCs are absolutely critical for synaptic function because they are the mechanism through which synaptic vesicles are activated and released at the active zone in response to an action potential. There's also calcium channels that directly funnel extracellular calcium into the ER, and this bypasses the cytosol. These are called store-operated calcium channels, or SOCs, or SOCs for short. They are critical for stockpiling calcium into the ER. Remember, I said calcium is very high inside the ER, and that's mainly because of these store-operated calcium channels. We'll see in a bit that the ER actually needs calcium to function properly in order for proteins to fold correctly. A lot of the folding chaperones are calcium dependent, so calcium needs to be high in the ER. The most commonly studied SOC is formed from the interaction between the plasma membrane-bound ORI1 channel and the ER-bound STIM1. Basically, calcium depletion in the ER causes STIM1 to oligomerize. So this is the oligomerized STIM1, and oligomerized STIM1 clusters at membrane ER contact sites where they bind with ORI1. ORI1 channels bound to STIM1 can then lead to the influx of calcium directly into the ER. Again, this bypasses the cytosol and allows the endoplasmic reticulum to accumulate cytosol, and that allows it to function properly. Let's also note these PMCA and NCX channels. I don't have NCX channels on here, but PMCA over here. PMCA is a P-type ATPase transporter, which is of the same class as a sodium-potassium pump, but instead PMCA simply uses ATP to efflux calcium. So PMCA is also extremely efficient at pumping calcium out, which means this is pretty much why calcium concentration is in the micromolar range in the cytosol. This is because PMCA is extremely efficient at using ATP to pump out calcium. PMCA is actually activated by calcium. Specifically, it's activated by calcium-bound calmodulin. Calmodulin we're going to be discussing a lot, but it's basically a small calcium-binding protein that, when bound to calcium, can modulate many different proteins. It's simply a calcium sensor. And one of the important proteins that calmodulin activates includes this PMCA calcium uniporter. So it activates PMCA and it uses ATP to efflux calcium. In more serious scenarios of calcium influx, if there's too much calcium in the cell, it activates NCX channels. The N is for sodium, the C is for calcium, and the X kind of looks like a transporter, maybe, I guess. It kind of looks like it's transporting two things. Anyways, the NCX imports 3 sodium atoms in exchange for exporting a single calcium ion. And it's typically only utilized during very high intracellular calcium conditions, but it's also extremely efficient at removing calcium. The downside is that there's an influx of sodium, which inhibits and blunts action potentials, and so that requires sodium to be pumped back out of the cell by the sodium-potassium pump. So NCX channels are energetically very expensive, but they're worth it if a cell is experiencing excitotoxic conditions. If there's too much calcium in the cell, they go out through the NCX channels. It's kind of an emergency mechanism of getting rid of calcium by utilizing the sodium gradient. Sodium is high outside the cell and it wants to come in, and it can do that to export calcium if situation requires it. So to summarize this slide, calcium enters the cell primarily by TRPC ligand-gated ion channels like MMDAR, or voltage-gated calcium channels, VGCCs, or directly into the endoplasmic reticulum through these ORI1 and STIM1 interactions. Since the ER is hoarding calcium, it's probably not surprising to learn that the ER can regulate the release of calcium as well. The ER can shoot out these little calcium puffs primarily by these IP3R channels, IP3s, and also RYRs, or ryanodine channels. And IP3Rs are activated by IP3. It's IP3 receptor, so it's activated by IP3. And IP3 is the head group of phosphatidylinositol phospholipids. So on the next slide, we're going to talk about that pathway. The ryanodine receptors are also important sources of these little calcium puffs that are getting shot out of the ER. And the RYRs basically function to augment and modify calcium transients. I think of them as kind of fine-tuning the calcium transients occurring in the cell, and they kind of depend on the cell type. So in terms of modulation, these IP3Rs and RYRs are primarily modulated by cyclic ADP ribose, which is basically cyclic AMP, but with a ribose sugar attached. So they're called C-ADP ribose. They can also be regulated by calmodulin. They can be regulated by AKT-mediated phosphorylation, AKT, and PML. We're going to see these examples in the apoptosis slide, but we'll talk about that later. But the basic idea is that calcium flux at these mitochondrial associated membranes primarily occur through IP3Rs. And calcium flux into the cytosol primarily occurs through these TRPCs, NMDARs, or AMPA receptors to voltage-gated calcium channels, and a couple others. So on the next slide, we're going to be talking about calcium signaling in the context of mitochondrial associated membranes. This is just a really quick overview of what we talked about in the last slide. We have LTCCs. These are also called voltage-gated calcium channels. We see our TRPC, activated by diacylglycerol. Diacylglycerol likes to activate TRPCs. We see IP3. Let's see, IP3 right here. IP3 is derived from the phospholipase C cleaves PIP2. Phospholipase C cleaves PIP2, which releases IP3. And IP3 diffuses to the IP3 receptor, IP3Rs, on the endoplasmic reticulum that releases calcium. And finally, we see that some forms of calcium regulation over here on the left. Protein kinase C, activated by diacylglycerol and calcium. Calmodulin, of course, and cyclic ADP ribose groups. Cyclic ADP ribose groups are derived from CD38. CD38 uses NADH and uses NADH, I think, for its ribose group, and it produces cyclic ADP ribose. There's a couple of other ways that IP3Rs can be regulated. We'll talk about that later, but these are just this is kind of just a supplementary slide before we dive into calcium signaling. In this slide, we're really going to dive into some of the calcium-sensitive signaling networks. And the source of much of the cytoplasmic calcium is the ER. And this is going to be our ER. This is our ER lumen up here in the top. And this is going to be our cytoplasm. Everything down here is going to be our cytoplasm. So gating the release of ER calcium is going to be our IP3R. IP3R. And according to this paper from which the graphic was derived from, a primary mechanism of IP3R regulation are the classic BH3 domain proteins. BH3 domain proteins. So this pathway that we're about to go through right with these two guys is going to relate a lot to apoptosis. So if you're not familiar with apoptosis, I recommend looking into that before watching this presentation. But basically what's happening is these BCL2 proteins embedded in the ER membrane are constitutively inhibiting IP3Rs. BCL2 and BCLXL are constitutively inhibiting or at least dampening the ability for calcium to be released. But BH3 domain proteins, like BAX or BAC, BH3 domain proteins are binding and inhibiting BCL2. So these guys inhibit BCL2. So this is a double negative. If BAC and BAX are inhibiting BCL2, then there's a loss of inhibition on IP3Rs. So BH3 proteins are upregulated during stress or apoptotic conditions perhaps, and they inhibit BCL2, and this frees up IP3Rs, and it actually makes them more sensitive to IP3, the head group from PIP2. And this is an interesting mechanism because it relates apoptotic BH3 protein expression with calcium release. And if I had to make a personal judgment as to the most important mechanism of IP3R regulation, I personally think this is a really interesting and important topic. So again, if you're not familiar with BH3 proteins, I suggest checking out my lecture on apoptosis. But the basic idea is that during basal conditions, IP3R receptors are mostly constitutively inhibited by BCL2. Normally this is happening. But during apoptotic conditions, BAC and BACs come through and they inhibit BCL2. And this increases IP3R sensitivity to IP3, potentially leading to cytotoxic calcium flux. So for much of this lecture, I'll be discussing how calcium flux mediates apoptosis. But before I do that, it's important to recognize that calcium flux is also a physiologically normal event, and it's good for a cell. Calcium is incredibly important for long-term potentiation and memory formation and synaptic vesicle release and many other things. So while we're discussing calcium as a mechanism of inducing cell death, just remember that we're talking about massive, sustained, and aberrant calcium flux, not physiologically normal calcium flux. So this graphic dates back to 2009, so it's a little bit old at this point, but it still highlights many of the important calcium signaling pathways. So for example, I want to mention a couple of these. Calpains. Calpains are calcium-dependent proteases that contain a calcium-sensitive domain that is highly similar to calmodulin. So calpains contain a subunit that looks like calmodulin, and not surprisingly, this domain can bind calcium. And when it does, when calcium binds to the calpain, calpains are activated. They begin cleaving various proteins like neurofilaments. They cleave caspases into active caspases, and they can cleave ion channels, and they can cleave BID. BID is another BH3 protein that can inhibit BCL2, and many other things. So generally, calpain activation by calcium promotes apoptosis. Calcineurin. Calcineurin, we're going to be talking about a lot. Calcineurin is a calmodulin calcium-dependent phosphatase that promotes apoptosis. And it likely does this by removing the phosphate on BAD. Calcineurin removes an inhibitory phosphate on BAD. BAD is another BH3 protein that can inhibit BCL2. And it can do this not just on the ER membrane, of course, it can do it on the mitochondria. Mitochondria contain BCL2 proteins as well. Let's see, another important protein over here is DRP1. DRP1 is also activated by calcium, and DRP1 is a GTPase that mediates mitochondrial fission. It's a GTPase that oligomerizes. It can oligomerize around a mitochondria, and it can wrench together so tightly that it causes the mitochondria to split in half. So it's responsible for mitochondrial fission, and it's activated by calcium. So if there's aberrant calcium flux, we're going to have fragmented mitochondria. The last thing in this graphic I wanted to mention is DAPK1, DAPK1. So DAPK stands for death-associated protein kinase. DAPK is a calmodulin calcium-dependent kinase that promotes cell death. And the mechanism still isn't fully understood. It has something to do with P53, but other than that, it's not understood. But it is known to be a potent tumor suppressor because it activates cell death, so a cancer cell would want to get rid of it. And its overexpression causes cell death. But it is calcium dependent, and that's what's important for our discussion, is that this calcium dependent DAPK protein can induce cell death. And finally, note this very direct line between calcium and the mitochondria. The IP3R can release calcium sometimes directly to the mitochondria, where it activates a protein called MPTP. MPTP. This is the infamous mitochondrial permeability transition pore, MPTP. And we'll discuss it in a moment, but the summary, before we go on to discuss that, the summary is that there's these pro-apoptotic BH3 domain proteins like BAX and BAC and BID and BAD. And these guys can all inhibit BCL2. And this sensitizes IP3Rs to IP3 and thus calcium flux. So calcium flux can then signal through many different proteins. The four that I mentioned are the calpanes, class of activated calcium-dependent proteases. Calcineurin is a phosphatase capable of activating BAD, a BH3 protein. DRP1, a GTPase that induces mitochondrial fragmentation, and DAPK1, death-associated protein kinase, strongly associated with apoptosis. And ultimately, calcium signaling leads to changes in gene expression as well. So this is kind of focusing on how it influences mitochondria, but these guys can also go to the nucleus. So just remember that this influences calcium signaling in the nucleus as well. And we're going to talk about MPTPs a lot, I think in the next slide, but very important as well. Now let's talk a little bit about mitochondrial ER contact sites. So the harbinger of calcium signaling, the main cause of calcium signaling, is the activation of phospholipase C, PLC, phospholipase C. And phospholipase C is typically activated by the alpha subunit of a GPCR. So there's many different ligands that can activate many different GPCRs, and a lot of them converge in the activation of phospholipase C. PLC cleaves PIP2 into diacylglycerol. Diacylglycerol is the phospholipid that sorry, not a phospholipid, it's diacylglycerol, but it's hydrophobic, so it remains in the intramembrane space. It also creates IP3. IP3 will then diffuse to these IP3Rs found primarily on mitochondrial associated membranes, MAMs. And one of the most important activities going on at these mitochondrial associated membranes is calcium transfer from the ER to the mitochondria. Remember, we said mitochondria generally have very low calcium concentrations unless stimulated. Well, calcium stimulation in mitochondria leads to increased ATP generation. We see increased ATP generation. This happens because calcium binds to numerous citric acid cycle, TCA enzymes and various electron transport proteins, and increases their efficiency. So increased ATP concentration means less AMP, right? You have less AMP, and if you have less AMP, you're not going to have activated AMPK. This increased ATP means less activated AMP kinase, which is a kinase that depends on AMP concentration. And this ultimately leads to a regular basal level of autophagy. And I have a slide coming up on calcium's regulation of autophagy. That's kind of the general idea. However, too much calcium flux, if you have too much calcium flux, it leads to depolarization of mitochondria due to the opening of these mitochondrial permeability transition pores, MPTPs. MPTP is the name of a yet to be identified complex that leads to the release of matrix molecules larger than 1.5 kilodaltons. Current theories suggest the MPTP consists of VDAC, ANT, ATP synthase, and cyclophilin D. Cyclophilin D is the only protein that's definitely involved in the MPTP, but the others are implicated but not yet confirmed. But whatever is mediating this MPTP, it appears to be dependent on calcium, right? The MPTP appears to be dependent on calcium flux, and when it opens, it leads to the efflux of protons, so the loss of a proton gradient, and also the release of cytochrome C. Cytochrome C is basically synonymous with apoptosis when it's released into the cytosol. And this leads to a loss of ATP production, right? Because if you lose proton gradient, you lose ATP production. That means AMPK is going to be active, and that means that there's going to be activated autophagy, and autophagy is going to be trying to salvage ATP concentration. Additionally, some of these MPTP channels are permeable to water. So water can actually flow into the mitochondria, can diffuse into the mitochondria, and this water influx can actually rupture the mitochondria from osmotic pressure. So calcium is both essential for mitochondrial health and it's also essential for cell death, depending ultimately on the concentration of calcium. So a careful balance of calcium is critical for mitochondria. And since mitochondrial calcium flux is actually derived from the endoplasmic reticulum, we're beginning to realize how important these MAMs are. So in this image, you can see the regulation of IP3R receptors by a bunch of different players. P53, when it's been exported from the nucleus, it can interact with IP3Rs. I'm not exactly sure how or what the effect is. FBOX XL2 is a ubiquitin ligase that triggers the degradation of IP3Rs. FBOX XL2 is a ubiquitin ligase. PML, we're going to talk about that in a second. And AKT, there's a bunch of different ways that these IP3R receptors are regulated. So in the next slide, we're going to zoom in at a typical mitochondrial associated membrane and look at some of the regulation and various events that are happening at the MAM. This is the current model of what an ER mitochondrial contact site actually looks like. First, note that the actual tethering between the two membranes is mediated by these MFN heterodimers. So you might recognize MFN. MFN stands for mitochondrial fusion. It's the protein responsible for mediating the fusion of mitochondrial outer membranes. So MFN 1/2 interactions between mitochondria mediates membrane fusion, while MFN 1/2 interactions between the mitochondria and the ER membrane simply functions to tether the mitochondria to the ER membrane. Inside the ER, we see these two proteins called calreticulin and calnexin. These are proteins with the ability to bind to hundreds of calcium ions. You see just a few of them right here, but these are calcium ions, and these two proteins bind hundreds of calcium ions, earning their reputation as calcium buffers. So much of the calcium inside the ER is bound to various folding chaperones like calreticulin and calnexin, and perhaps not coincidentally, calreticulin and calnexin tend to be concentrated at these MAMs, these mitochondrial associated membranes. Calnexin is localized to MAMs through the activity of this cytosolic PAX2 protein. PAX2 is in fact involved in actually localizing many other proteins to the mitochondrial associated membranes. It's kind of thought of as the coordinator of the construction of a MAM. Loss of PAX2 causes severe intellectual disability in humans. And in cell culture, these cells lacking PAX2 don't have mitochondrial associated membranes. And they also have dysfunctional and highly hyperfragmented mitochondria. So PAX2 is thought to be involved in coordinating the construction of this entire complex by recruiting the necessary proteins. The actual calcium channel itself is formed from the linkage of the IP3R, with VDAC. VDAC stands for voltage dependent anion channel. And this IP3R VDAC complex is nucleated by the GRP75 folding chaperone, GRP75. siRNA-mediated knockdown or knockout of, or to be knocked down of GRP75 abolishes the functional coupling between these two receptors. Another important player at these MAMs is the PML protein, PML. You might recognize this protein as a critical tumor suppressor protein that, when lost, can promote the development of promyelocytic leukemia, which is why the protein's named PML, for promyelocytic leukemia. So this protein promotes apoptosis, and it's thus a tumor suppressor protein. And specifically, it does this by binding IP3R. Or it promotes apoptosis by binding to its IP3R, and it facilitates the transfer of calcium into the mitochondria. The loss of PML, as you might see in a cancer, leads to the loss of calcium transfer, and this leads to the inhibition of apoptosis. It also leads to the upregulation of autophagy due to imbalances in the ATP to AMP ratios. Remember, mitochondrial calcium is important for metabolism and ATP generation. And so the loss of calcium leads to deficits in ATP, which activates autophagy by AMPK. So PML is actually a very interesting protein because it helps induce apoptosis but can also influence autophagy. And lastly, there's the sigma-1 receptor. Sigma-1 receptor. These proteins are co-localized with IP3Rs, and they appear to prevent the ubiquitination and degradation of IP3Rs, thus prolonging calcium transients. So to summarize, we have MFN proteins tethering the two membranes. We have GPR75, which is bridging the IP3R and the VDAC channels. And we have PML and sigma-1 receptors that are stabilizing the complex. We have calreticulin and calnexin. These are providing the calcium that is transferred. Then we have PAX2, which directs components like calnexin to the correct location in order to construct this entire complex. The last player is MCU. MCU, which stands for mitochondrial calcium uniporter. It's the inner mitochondrial membrane calcium channel. When this channel was first discovered a couple of years ago, it generated a lot of interest because based on everything we know about mitochondrial calcium, it should be incredibly important, right? It is presumably how calcium gets into the matrix. Except, unfortunately, knockout studies have shown us that MCU is dispensable. MCU can be knocked out, and the mice only show defects in skeletal muscle. They have about 15% less performance capacity, which doesn't make any sense to me personally. And it was kind of a letdown for the scientific community. If MCU is the channel through which calcium enters the mitochondria, then why doesn't knocking it out cause severe dysfunction? Instead, MCU knockouts appear to have little effect on mitochondrial respiration capacity. So calcium flux into the mitochondria might not be as important as we think it is for ATP generation, which is at odds with what I've been telling you so far. But that's the nature of science, conflicting reports. It is instead possible that calcium flux into the intermembrane space perhaps is sufficient for calcium-induced ATP generation, which would explain why MCU is dispensable. Anyways, don't be disparaged by this because mutations in various proteins like NCX, there are NCX transporters on the inner mitochondrial membrane. Remember, these proteins are responsible for removing calcium from the matrix. They export calcium, and mutations in NCX causes it's a highly lethal mutation. Additionally, the MICU1, the MICU1 protein, which functions as a negative regulator of MCU, is also incredibly important. Loss of function mutations in MICU1 result in constitutively active MCU1 channels, leading to severe mitochondrial dysfunction and ultimately causes intellectual disability with progressive myopathy. So the theory at this point is that calcium transients into the mitochondria may not be very important for ATP generation, may be dispensable. But calcium overload is still a very relevant and toxic process, because if that process is disturbed by preventing calcium efflux through NCX channels or deregulating MCU and allowing constitutively active calcium import, those are two very highly toxic events. So it's still very relevant in terms of how much calcium is getting into the mitochondria, because that's a very toxic process. We're going to discuss that a little bit more in the next couple of slides. This figure gives us another close-up of the mitochondria, showing us many of the relevant players in calcium transfer. So calcium enters through the VDAC channel, which is linked up with MCU. right? Mitochondrial calcium uniporter. And between we have these MEQ1 and MEQ2 regulatory proteins. They regulate MCU calcium transfer. Our MFN proteins are present, and these are responsible for tethering our mitochondria, perhaps to the ER, or mediating fusion of the outer mitochondrial membrane. We have another interesting protein, MIRO, MIRO. MIRO is a protein that functions as the adapter to kinesin or kinesin, and so MIRO is responsible for mediating anterograde transport along microtubules. So if a mitochondria is moving down an axon to the synapse, it's probably because MIRO is linking up with kinesin. Kinesin would be right here, then you'd have our microtubule. We also have DRP1. DRP1, remember, is a calcium-dependent GTPase that mediates mitochondrial fission. So this GTPase actually binds to various receptors on the mitochondria, including MID49 and FIS1, FIS for fission. And the dependence on calcium means that if there's overloaded calcium, we can see increased mitochondrial fragmentation because of this DRP1 interaction with its receptors on the mitochondria. See, so healthy calcium flux, it's right here we see a green arrow. This is indicating healthy calcium flux, and this activates various dehydrogenases to generate NADH and is good for our cell, right? We also see OPA1 right here. OPA1 sitting here at the neck of the cristae will be important for our next slide, but basically OPA1 functions to fuse the inner mitochondrial membranes during mitochondrial fusion. And it's also responsible for sequestering the contents of cristae, right? This is our cristae down here, these little sacs that kind of protrude into the matrix. That's our cristae. And OPA1 is responsible for sequestering the contents of the cristae through an unknown mechanism. However, during calcium overload, an unknown calcium-sensitive protease actually cleaves OPA1. A calcium-sensitive protease cleaves OPA1, rendering it non-functional, and this causes the cristae to open up, allowing the release of what's in sequestered in these cristae. And one of those things can be cytochrome C. Calcium overload also activates our MPTP, which may or may not include the F0, F1 subunit of ATP synthase, which is another interesting facet that we probably don't have time to go through. And finally, calcium overload activates our NCX transporters. This is our NCX transporters, and they will basically burn ATP trying to remove calcium from the matrix. The problem is if our MPTP is open, calcium is going to be, or sorry, our calciums can be rushing in no matter what. And so the activation of the NCX transporter is a futile cycle. It's transporting calcium for calcium just to rush back in or rush back out. The membrane's been permeabilized, so the transporter is wasting ATP to try and regulate the concentrations of sodium and calcium. So this NCX transporter is actually burning ATP. So this is a pretty exciting slide. We're going to be discussing how calcium flux influences cell death. So in panel A on the left, we have typical basal healthy conditions, right? Our IP3Rs are constitutively inhibited by BCL2 and BCLXL, maybe not inhibited, but they're desensitized to IP3. We also have our IP3Rs phosphorylated by AKT, which has been found to dampen calcium transfer during apoptotic conditions. Our apoptotic proteins, APAF1 and caspase-9, are not active. They're inactive in the cytosol. And our cytochrome C down here is sequestered in the cristae, in the sacs of the cristae. However, during apoptotic conditions, BCLXL has been displaced. It has been displaced by the upregulation of BAX and also BAD, but primarily BAX. And this sensitizes our IP3Rs to IP3, right? In addition, our PML protein, via some unknown regulatory mechanism, has recruited and then inhibited AKT. PML inhibits AKT at these MAMs. Remember, AKT placed phosphorylations on IP3R that attenuated its activity during apoptosis. PML simultaneously recruits the PP2A. PP2A is a phosphatase that removes inhibitory phosphorylations from IP3R. It also recruits P10. You've probably seen P10 before. It's a very commonly mutated protein in cancers. And so PML, the two big events that happen during apoptosis are the loss of BCLXL or BCL2 due to the presence of BAX. BAX displaces these interactions. And we see the loss of inhibitory phosphorylations. We see the loss of this phosphorylation due to the inhibition of AKT and the recruitment of a phosphatase that specifically removes that phosphorylation. When this happens, it leads to GRP75 linking up the IP3Rs with VDAC, and they begin facilitating calcium flux into the mitochondria. This activates cristae remodeling because we see calcium-dependent cleavage of OPA1. It's not shown here, but OPA1 would be right here, right? And OPA1 then gets cleaved when calcium rushes into the cell, and this remodels the cristae and allows them to open up. OPA1, which is also critical for mitochondrial inner membrane fusion, is now cleaved, and it's no longer able to mediate the fusion of mitochondrial membranes. So the depolarized mitochondria at this point is going to be cut off from the rest of the mitochondrial reticular network. So this also leads, obviously, to cytochrome C release, and it's speculated that this might also permeabilize the mitochondrial membrane to calcium. In this, if the mitochondrial inner membrane has been permeabilized to calcium, this bypasses the role of MCU. Regardless, calcium flux into the matrix, mitochondrial calcium transfer into the matrix activates the PTPs or MPTPs, and this leads to the loss of the electrochemical proton gradient and thus ATP generation. The cytochrome C that escapes is able to activate executioner caspases like caspases 3 and 9, and it does that through caspase 9. Right? Cytochrome C recruits caspase 9 and activates caspase 9, which then cleaves and activates caspases 3 and 7. So there's a lot going on. The basic pathway is this: BAX upregulation, perhaps due to BID or some other BH3 protein that is binding and inhibiting a BCL2 or whatever. promotes IP3R activation by removing or binding to IP3Rs, right? Binds to IP3Rs and increases their sensitivity to IP3. PML is also activated, and it drags AKT away from IP3Rs and inhibits it, and simultaneously recruits PP2A phosphatase to remove inhibitory phosphates from IP3R. IP3Rs then link up with VDAC, right? We see the linking up between VDAC and the IP3R. We see the influx of calcium. Calcium leads to the cleavage of OPA1, which releases the cytochrome C. We also see the permeabilization of the membrane due to MPTP, which causes depolarization, dissipates the proton gradient. We see reactive oxygen species getting generated as a result, and this just leads to apoptosis. The cell is done at that point. Calcium flux can activate not just the apoptosome, but also this complex called the inflammasome. The inflammasome, as it sounds, is typically a response to infection and inflammation. The inflammasome is composed principally of NLRP3, which is similar in function to APAF1, meaning NLRP3 oligomerizes. NLRP3 oligomerizes and recruits a protein called ASC. So right here, this would be the ALC. Or sorry, not ALC, ASC. And ASC uses a CARD domain, a caspase recruitment domain, to recruit procaspase-1. It recruits procaspase-1 and activates it, and caspase-1 begins cleaving pro-IL-1 beta into activated IL-1 beta, which is then released to cause local inflammation. So what causes the oligomerization of NLRP3? Well, two main events drive inflammasome formation. Potassium efflux. You can see the potassium is leaving the cell right here. So potassium efflux leads to the activation of NLRP3 oligomerization. And also calcium influx. Calcium influx. So calcium overload in the mitochondria causes dysfunction. And it's not just apoptosis that can ensue, but also the release of mitochondrial DNA and reactive oxygen species. Calcium influx can occur by the classic IP3R pathway, as we just discussed, but it can also occur through these TRPM2 channels. TRPM2 channels, and these channels are actually sensitive to reactive oxygen species, perhaps fueling a positive feedback loop, meaning you have dysfunctional, damaged mitochondria that produce reactive oxygen species, opens calcium channels, which floods mitochondria with calcium, which causes more reactive oxygen species, and you get this positive feedback loop. So dysfunctional mitochondria release ROS, which open these TRPM2 channels, which causes even more calcium influx and calcium overload. Mitochondrial DNA and ROS signaling due to this calcium overload can then cause NLRP3 to oligomerize. The exact mechanism of how mitochondrial DNA and reactive oxygen species caused NLRP3 to oligomerize is not understood. But regardless, calcium is most definitely capable of destabilizing mitochondria. Right, a very simple pathway. Calcium-induced cellular dysfunction seems to revolve around aberrant calcium overload inside the mitochondria. So although cytosolic calcium signaling, out here, although it's important, it seems to me that the breaking point is when mitochondria are overloaded with calcium. mitochondrial calcium overload can induce apoptosome or even inflammasome formation. And I think the difference of which probably depends on cell type or maybe gene expression or other signaling events that are happening, for example. There's also evidence that calcium modulates autophagy. So calcium flux derived either from IP3Rs right here or even from lysosomal, these are called mCoL-N1 channels. Wherever the calcium comes from, it can activate KMK beta. KMK beta is a protein activated by calmodulin. So these calcium ions can indirectly activate KMK beta through calmodulin. And these calcium fluxes, when they activate KMK beta, they can cause KMK beta to phosphorylate and activate, AMPK. So AMPK can be phosphorylated and activated by KMK beta. And again, AMPK is that critical starvation activated kinase that is upstream of ULK1. And ULK1 is the initiation complex of the autophagisome. And so ULK1 requires two things. AMPK requires two things. It requires phosphorylation by KMK beta or liver kinase B1, which is another kinase. And AMPK also requires AMP. And cytosolic calcium is able to do both of these things. So cytosolic calcium activates KMK beta through calmodulin, and it also simultaneously upregulates AMP. Cytosolic calcium suggests that mitochondrial calcium buffering has failed and that the mitochondria are starved of calcium and thus they are producing less ATP, meaning AMP is upregulated and the AMP to ATP ratio is imbalanced. So if there's a high concentration of calcium in the cytosol, that means there's probably a low concentration of calcium in the mitochondria, and you don't have the correct ATP to AMP concentration. Let's also not forget calcineurin. Calcineurin in this graphic over here to the right is just CN. It's another calcium dependent or calmodulin dependent phosphatase. And one of the many proteins targeted by calcineurin is TFEB. TFEB is the master autophagic transcription factor. The only problem is TFEB is constitutively phosphorylated by mTORC, and this inhibits it because this phosphorylation recruits the serine threonine binding protein 1433. And this inhibitory phosphate can then be removed by calcineurin. And this allows TFEB to enter the nucleus and prime lysosomal and autophagic gene expression. So calcium flux activates autophagy in at least 3 different ways. We have KMK beta can be activated, which KMK beta activates AMPK, and AMPK is responsible for activating the autophagic initiation complex, ULK1. The pre-initiation complex, ULK1. And loss of calcium from the mitochondria, the efflux of calcium from the mitochondria decreases ATP generation, which also activates AMPK if you have imbalances in the ATP to AMP ratio. And you also have activation of calcineurin, which removes the inhibitory phosphate on TFEB, allowing it to enter the nucleus and prime gene expression. So there are a bunch of other proposed mechanisms of calcium influencing autophagy, but it gets exponentially more complex because there isn't any clear-cut pathway that's been identified. It's kind of all over the place. But I decided to highlight just these 3 mechanisms. But the ones that I thought made the most intuitive sense are CAMK beta activation, calcineurin activation, and imbalances in the ATP to AMP ratio. Okay, so one of the last topics I wanted to cover is calcium-activated gene transcription in brain cells, which can get really complicated really fast, so we're just going to stick to the basics. But basically, calcium enters the nucleus through simple diffusion, right? There's very large nuclear pore complexes. Calcium can diffuse right into the nucleus. And the calcium is typically derived from voltage-gated calcium channels. Since I'm a neuroscience guy, this means that action potentials trigger calcium transients by depolarizing the soma, and this opens the voltage-gated calcium channels. The second primary mechanism of calcium flux is, of course, our IP3 receptors. In order to study the effect of calcium in just the nucleus, researchers often used a calcium chelating molecule called BAPTA, BAPTA, and they would fuse BAPTA to dextrin. And this would make the molecule large enough as to where it couldn't diffuse out of the nucleus, but it was still effective at chelating calcium. They would do this, and they would microinject these BAPTA dextrin molecules into the nucleus, and then they would look at gene expression, because it would completely abrogate calcium flux in the nucleus. And it also completely prevented Krebs activation. So this calcium chelating molecule completely prevented Krebs-mediated gene transcription. Krebs, you might recognize, is a critical and very fundamental transcription factor in the nucleus of neurons, and it mediates long-term potentiation and and neuronal plasticity. And it does this by essentially functioning as a calcium-sensitive transcription factor. So the more depolarization events that occur, the more voltage-gated calcium channels are open, and the more calcium flux into the nucleus, and the more Krebs initiates gene transcription. And it does this by sensing calcium, although it doesn't do this directly. Krebs senses calcium through CAMK4. CAMK4 phosphorylates and activates both Krebs and Krebs binding protein, CBP. And when CAMK4 phosphorylates these two proteins, it induces gene transcription that is relevant to synaptic plasticity and LTP. Another calmodulin-dependent kinase is CAMK2. CAMK2 is also activated, and it's known to modulate histone or chromatin structure by activating, here it is, CAMK2 is known to activate chromatin structure by phosphorylating MECP2. MECP2 is a DNA methylation binding protein that functions to remodel chromatin. So two of the really important calcium-sensitive kinases are CAMK4 and CAMK2, both activated by calcium flux. Another important event is the binding of calcium directly to this protein called DREAM. DREAM stands for DRE antagonist modulator, and DREAM is simply a repressor of gene transcription, but owing to several EF hand motifs, DREAM can bind calcium. Remember, an EF hand motif is the motif that binds calcium. I didn't mention that, but that's important. And anyways, DREAM has multiple EF hand calcium binding motifs, and when DREAM binds to calcium, it detaches from DNA, allowing gene transcription. So when calcium flux enters the cell, enters the nucleus, it binds to DREAM and it inhibits the repressor. It causes DREAM to detach from the DNA. It's generally believed that calcium signaling through DREAM, CAMK4, and CAMK2 are all neuroprotective. So blocking neuronal calcium would be basically equivalent to telling the neuron that it's not synaptically active, which would probably lead to apoptosis or pruning. Paradoxically, some certain NMDARs can actually turn off intranuclear calcium signaling through Krebs, and this promotes cell death. So that's a strange process, and we're going to examine that in the next slide. Most NMDARs are found at synapses. Most NMDARs are found at synapses, and this is healthy, it's normal, and their signaling generally promotes nuclear calcium flux and cytoprotective gene transcription. Right. However, something goes awry when NMDA receptors are located extrasynaptically, so outside the synapse, on the soma perhaps, and something goes wrong when they get activated. Normally, glutamate isn't found extrasynaptically, and so this isn't a problem. But during neurodegeneration, or perhaps there's astrocyte dysfunction, glutamate actually gets out of the synaptic cleft, and it can activate these extrasynaptic NMDARs. And extrasynaptic NMDARs contain unique subunits and unique postsynaptic densities. And calcium flux at these locations ultimately lead to the activation, or they ultimately lead to the loss of Krebs signaling. And we just talked about how Krebs is very important for cytoprotection. And extrasynaptic NMDARs ultimately lead to the downregulation of Krebs, likely through the activation of this protein called Jacob. Jacob is a Krebs phosphatase. It's a Krebs phosphatase that will, when activated, it can translocate to the nucleus and remove phosphates from Krebs, or specifically Krebs binding protein, which inhibits Krebs. Extrasynaptic NMDARs also activate DAPK, death-associated protein kinase, and they also activate FOXO3. And FOXO3A is a pro-apoptotic transcription factor. So when it's activated by these extrasynaptic NMDARs, you see cytotoxic gene transcription, like BIM, FASL, FOXO1. In addition, extrasynaptic NMDARs tend to be closer to mitochondria, which is what I've depicted right here. So I added a little mitochondria, and thus it's speculated that their activation is mitotoxic by directly overloading them with calcium. So when it comes to NMDARs and neuronal calcium flux, the actual location of NMDA receptors is really critical, because extrasynaptic NMDARs are coupled to unique postsynaptic densities that can activate Jacob, for example, which inhibits Krebs. You can also see the activation of DAPK to promote apoptosis. And extrasynaptic NMDARs are also in close proximity to mitochondria to expose them to potentially harmful calcium flux. So although the nucleus itself cannot distinguish calcium flux that's derived from synaptic NMDARs versus extrasynaptic NMDARs, what likely makes a difference is the presence of extrasynaptic NMDAR activated proteins like Jacob or FOXO3A. And these proteins promote apoptosis. But the precise mechanisms of how these extrasynaptic NMDARs activate Jacob or FOXO3A isn't understood. But it's a very interesting phenomenon that the location of these ligand-gated calcium channels makes a big difference in terms of what's going on later in the nucleus. Anyways, that's about all I have to say for calcium. It's still a highly active field of research. There's a lot going on. And if you have any questions about any of these slides, please let me know. And thanks for watching.